Hello, everybody. This will be a simple introductory lecture about human embryology from Amr Zaher, the gentleman of medicine. Yeah, this is me. So, first question, can you imagine how the human baby develops? However, you imagine how the human heart or human baby develops, what is really going on is not like what you imagine. So let's start from here. The sperm. The sperm is the smallest cell in the body. It is a haploid cell, meaning it has a half number of chromosomes. It consists mainly of three parts, head, tail, and a midpiece in between. Here is the acrosome, the nucleus in the head, and the tail, and in between is the mitochondrial spiral. Here is something very weird have happened, that the mitochondria get out of cytoplasm to be outside. This is essential for the sperm as it uses for energy production for his long trip inside the female genital tract. Here is the place where the digestive enzymes are released, allowing the head to penetrate the female egg. And here is the haploid number, I mean 23 chromosomes. And here is the spiral that produce ATP, as I just mentioned, for the long trips in the female cervix, uterus, and uterine tube. And here is the tail that allow movement of the sperms. However, the movement of the sperms is not directed. They move everywhere. And only the lucky one who reaches the ovum is by chance. The ovum is the haploid cell. It is the largest cell in the body. It can be even seen by the naked eye. 0.2 millimeter in size. Here is the zona pellucida, corona radiata, nucleus cytoplasm, some details about the ova. This is the outer layer. Keep the sperms out. All the sperms, not for the lucky one. Corona radiata supplies protein and element for growth of the ova, help fertilization, and prevent entry of more than one sperm in most of the cases. And the nucleus with the 23 chromosomes. The sperm cells is haploid, so it has 23 chromosomes, join the other 23 chromosomes from the female egg to form diploid cell. Here is the entry of the lucky one to the ova. Here is the zona pellucida surrounding the zygote. Here is the formation of the first somatic cell. It is the zygote. And this is the beginning of life for all animals, from mice to elephant. And it is diploid cell containing in humans 46 chromosomes. And this number varies from species. And crossing over, mechanistic over, it is taking over. That's why John Kennedy looks like a brother for Edward Kennedy. This is not Edward Kennedy. This is John Kennedy and his younger brother. They look like brothers, but they are not identical because they're crossing over. And they both get killed by assassination. Robert Kennedy and his big brother, John Kennedy. Cleavage. Cleavage is series of mitotic cell division of the zygote resulting in formation of blastomere. First two cell stage and fourth cell stage surrounded by zona pellucida, eighth cell stage and morula, many cells. And this is, we can call it totipotent embryonic cells, not used for medical purpose. And a cavity will appear inside to form a structure called blastocyst. So blastocyst is a structure with cavity. This is the first cavity to be appear in the embryo. The inner cell mass will give all the embryo. This is in violet will give all the embryo. And the cavity here, the first cavity will be formed is the blastocoele. It surrounds the inner cell mass. Baby and embryonic cells here or stem cells here like water because it decreases the force of gravity on the growing cell and dividing cells. We need to decrease the force of gravity, so we like to form cavities filled of water surrounding the baby. Here again, the blastocyst, the outer cell mass, trophoblast, inner cell mass, this is the primordium or beginning of the embryo, blastocell fluid-filled cavity, surrounded by zona pellucida. Here is the embryonic pool that gives the embryo. So it will hatch, okay, get rid of zona pellucida, and to be implanted in the uterus. Here is the female genital tract. Here is a part of the female genital tract. Here is the layers of the uterus, endometrium, myometrium, perimetrium. The endometrium consists of a single layer of columnar epithelium plus the stroma, which is connective tissue. This columnar epithelium, and here is the stroma formed of connective tissue. Migration will start by the help of peristalsis of the uterine tube and motion of the cilia of the lining of the tube. Here is the fertilized egg, where the sperm meets the egg and fertilize it, and then the egg will move in the fallopian tube to go inside. As I mentioned before, the egg mitochondria is inside, so the egg cannot move by itself. And of course, it has no tail. So it moves by the help of the cilia and peristaltic movement of the fallopian tube. And here it reaches the uterus, the inner cell mass, trophoblast, blastocyst cavity, come closer to the columnar epithelium and start to invade the posterior wall of the uterus. This is the usual site where the embryo implanted. Of course, it can be implanted anywhere, but this is the usual course of implantation. The trophoblast cell as the embryonic pool will divide and invade the uterus. Here, the trophoblast will divide and invade the uterus. Invade again, and a collection of trophoblastic cells will collide together to form syncytial trophoblast, as it showed here. What is syncytial trophoblast? It came from our syncytium, mean a mass of cytoplasm containing nuclei not surrounded by cell wall. Here is the inner cell mass. It will divide it again into hypoblast and epiblast. Hypoblast in yellow, epiblast in blue. Great. Epiblast and hypoblast. Hypoblast cuboidal cells, while epiblast columnar cell. This is the first embryonic disc. We call it bilaminar embryonic disc because it consists of two layers. Here in concrete, I'm holding it in my hand, bilaminar embryonic disc. Invasion continue, like you see here. And hypoblast is ventral, while epiblast is dorsal. Hypoblast forms the upper layer, while epiblast forms the lower layer. I know that embryology is a little bit confusing, so we always have to remember these new terminologies, like syncytial trophoblast, cytotrophoblast, endometrium, stroma, hypoblast, and epiblast. Implantation will continue. While implantation continues, another cavity will appear within layer of epiblast. This will be the amniotic cavity. The dividing cell asks for water to decrease the force of gravity on them. So another layer was formed, amniotic cavity. Okay, the amniotic cavity starts small, but eventually at the end of pregnancy, it will enclose the whole embryo, except in one place. Yes, you're right, the umbilical cord. Syncytial trophoblast will cover more and more of the blastocyst. Remember, syncytial trophoblast, cytotrophoblast, amniotic cavity, blastocyst cavity, epiblast, and hypoblast. Implantation continue, and the syncytial trophoblast will surround the whole future embryo. Okay? And coagulation plaque will seal the small hole where the blastocyst implants. Here the implantation is completed. within the uterus and embedded in the endometrium here. And here is some details I'm not going to mention here in this lecture. There's more details in the coming lectures. The amniotic cavity is expanding. At the same time, syncytial trophoblast expanding, surrounding completely the embryo. And trophoblastic lacuna will come and settle down in syncytial trophoblast. It's full of mother's blood. Great. Cells from the hypoblast start to migrate in two waves of migration. The first wave of migrations, as it's shown here, will completely cover the inner surface of cytotrophoblast and form Heather's membrane, or we call it exocelomic membrane. So Heather's membrane is formed of hypoblast cell and extracellular matrix. At this point, the primary sac replaces the blastocystic cavity. We change names. After the first wave of migration, we change the name from blastocystic cavity to primary sac. And the difference is the cavity on the left is surrounded by cytotrophoblast, while on the right, surrounded by hypoblast in yellow. The extraembryonic mesoderm is expansion between the primary yolk sac and cytotrophoblast. Cytotrophoblast is shown here in gray color. What is it? What is the extraembryonic mesoderm? Some of migrating hypoblast cells make transdifferentiation into mesenchymal cells that fill the space between Heather's membrane and cytotrophoblast, forming the extraembryonic mesoderm in gray here. And the trophoblastic lacuna will become filled of maternal blood from them. The extraembryonic mesoderm expand between the amniotic cavity and cytotrophoblast. Look, complete expansion between of the extraembryonic mesoderm. The extraembryonic mesoderm will split in two layers, one coating the outers of Heather's membrane, while the other lining is inside of the cytotrophoblast. And another cavity will show up, the chorionic cavity. Let me introduce to you the chorionic cavity, the third cavity, fill of fluid. So here we have amniotic cavity, primary yolk sac, and chorionic cavity. Fill of fluid to decrease the force of gravity, as I mentioned before. The second wave of migrating hypoblast cell, okay, will push the primary yolk sac in front of it. Will push again the primary yolk sac and replacing it by secondary yolk sac. So here is amniotic cavity, secondary yolk sac, chorionic cavity, primary yolk sac. And the primary yolk sac will disappear. So we have again three yolk sac. As the primary yolk sac will go away, we have amniotic cavity, secondary yolk sac, and chorionic cavity. Again, here is the amniotic cavity and secondary yolk sac surrounding the bilaminar embryonic disc. Again, here is the amniotic cavity, secondary yolk sac. Here is the epiblast, hypoblast. Together they name bilaminar embryonic disc. The bilaminar embryonic disc is suspended in the chorionic cavity by a thick connecting stalk. Here is the connecting stalk. Okay, and this is the prototype or promedium of the future umbilical cord. Here is the stalk. Here's syncytial trophoblast, extraembryonic mesoderm in gray, cytotrophoblast, and chorionic cavity. And of course, this is the three cavities: amniotic cavity, secondary yolk sac, and chorionic cavity. And the lacuna filled with maternal blood increases. I'll take this out for more details. This is syncytial trophoblast, cytotrophoblast, and extraembryonic mesoderm. Here is amniotic cavity, secondary yolk sac, and all are surrounded by chorionic cavity. Gastrulation. The embryo is sandwiched between two cavities, the amniotic cavity and the yolk cavity. Amniotic cavity and yolk cavity, or second yolk cavity. And it's all surrounded by chorionic cavity. Beautiful. And here is connecting stalk, the future umbilical cord. And here is the embryo embedded in the uterus. The extraembryonic coelom including the yolk cavity, amniotic cavity, and chorionic cavity. This cavity located outside the trilaminar embryonic disc, so they are called extraembryonic coelom or cavity. The intraembryonic coelom are cavity that occurs within trilaminar disc. Here is amniotic cavity and yolk sac, and the embryonic disc is in between. Cells within the embryonic disc must differentiate and migrate, forming three germ layers: ectoderm, mesoderm, and endoderm. This cell movement that establishes the primary germ layer called gastrulation, and the embryo during this period of time is called gastrula. The epiplast has now differentiated into three germ layers of the embryo. So the bilaminar disc becomes trilaminar disc. Again, remember, chorionic cavity. We have connecting stalk, syncytial trophoblast, cytotrophoblast, extraembryonic mesoderm. The three germ layers that will give the embryo all derived from epiplast, the blue. Some books call the epiplast the primitive ectoderm and the hypoplast the primitive ectoderm. Here is the amniotic cavity and second yolk sac. We'll get a cut section here. So the epiplast will be above and hypoplast below. Here is the same in concrete. I'm holding it in my hand. Hypoplast in yellow, epiplast in green. Surrounded by the amniotic cavity and secondary yolk sac. Bilaminar embryonic disc, holding it in my hand. So we'll make a cut section or transverse plane here in the bilaminar embryonic disc. We can identify a structure called primitive streak. It is elongated groove, bulk, with bulging sides in the epiplast, here in concrete. And a primitive node, it is a bulk, and with a middle depression called primitive bite, here. A primitive groove in the middle of the primitive streak, another structure. And we have a cranial end of the primitive streak and caudal end. So axes of the embryo are defined by the primitive streak. This is the first axis of the future embryo, as it has a caudal end and a cranial end. And a cut section here, and the primitive streak is shown here. Primitive node, and its bite with the primitive streak. Cells from the epiplast will move into primitive streak in epithelial mesenchymal transition. Epithelial cell will transform into mesenchymal cell and leave the basement membrane to go freely. A behavior is like what cancer cells do. Epithelial cells become mesenchymal cells, a multipotent, strong stromal cell that differentiate into all cells of the body. And it falls down here and go through the primitive streak in two waves of migration. The first wave of migrating cells will displace and replace hypoplast cells to find the definitive or the final endoderm. Here, it will replace here and forming the definitive endoderm. The second wave of migration will form a layer between ectoderm and endoderm. This is the mesoderm. So the three germ layers now are formed: ectoderm, mesoderm, and endoderm. And the embryo becomes trilaminar disc embryo. And another axis will form, the dorsal axis towards the ectoderm and the ventral axis towards the endoderm for the future embryo. So bilaminar disc is now trilaminar disc, or we call gastrula. Here is the bilaminar up on the top, and the and trilaminar disc is on the bottom, ectoderm, mesoderm, endoderm. And here is in concrete with these beautiful colors. I imitate the same colors. Ectoderm and endoderm and mesoderm in between. The ectoderm will give some structure like the enamels of the teeth, the nervous system, big part of the skin, epidermis, near and hair, subcutaneous gland, lenses of the eye. The mesoderm will give the heart, the muscle, the bone, blood cells, fat cells, spleen, blood vessels, serous membranes of the lining of body cavities, and the gonads. Mesoderm is a great step in evolution. It allowed more highly evolved organisms to have internal cavity that can hold and protect organs because it is the layer in the middle. Mesoderm is a germ layer that distinguishes a higher form of life. We call it bilaterally symmetrical life from lower life forms, radially symmetrical life. What is the radially symmetrical life or organism? Such organism has no left or right, they only have top and bottom surface. Like these two organisms you are familiar with, jellyfish and starfish. Of course, jellyfish is not fish and it's not made of jelly, nor the starfish is a star or a fish. While bilaterally symmetrical organisms can divide it by midline in two mirror image, only from outside, but internal organs can differ. Here is a bilaterally symmetrical organism. His left side from outside is like his right side. Ear, nose, skin, tongue. He is bilateral. Have on each side limbs, two limbs on each side. The endoderm, the inner layer will give the epithelial lining of urinary bladder, urethra, thymus, thyroid, parathyroid, gallbladder, pancreas, liver, epithelial lining of gastrointestinal tract. Here is the trilaminar disc with dorsal and ventral side, ectoderm, endoderm with mesoderm in between. Here again, the trilaminar disc surrounded by amniotic cavity, chorionic cavity, second yolk sac, and intraembryonic mesoderm is different than extraembryonic mesoderm as it's shown here in colors. So we'll come to the end of this brief lecture to meet another lecture from me. And the more I know, the less I know. And the less I know, the more I think I know. This is from Amr Zahir, the gentleman of medicine.